The 2019 ATS ERS spirometry standards have been updated to help alleviate the issue of suboptimal inhalation, which was previously not specifically considered within the assessment of lung function quality. In order to assess the impact of this new grading, the authors undertook a review of 646 measurements from 162 patients in the first 4 months of a clinical trial in asthmatic patients using the ATS ERS 2009 criteria. By means of a comparison, we evaluated the same measurements according to ATS ERS 2005 standards, and we've reported the incidence of suboptimal inhalation errors in efforts that would otherwise have been graded A or B using the old criteria. We identified 83 measurements, 12.8% of the total, which would have been rated acceptable and repeatable using ATS ERS 2005, which had a clearly determined suboptimal inhalation error according to the new criteria. These measurements came from 28 operators at 39 sites, which represent 58.3% of the operators and 43.8% of the sites. The FIVC minus FVC comparison showed forced exhalation started from an average of 271 milliliters below full inflation. The difference ranged from 106 to 722 milliliters, representing an average of 8.9% of the FVC, with a range of 5.1 to 18.4% of FVC. A common finding was the apparent absence of even the start of an inspiratory plateau on the initial inhalation, which suggests that many of these errors represent a coaching error with a possible instruction to the patient to blast out prior to observing full inflation. Repeatability of the two largest efforts has long been considered evidence that the efforts have started from full inflation. It is clear that repeatability cannot be used as a surrogate measure to show full inflation. Within the clinical research field, the presence of suboptimal inhalation, particularly at the start of trials, often results in the effect of suppressing baseline patient values, which then generates inflated treatment effects at a patient level and increased overall variability of the data at the study level. Adoption of ATS ERS 2019 standards should improve overall variability. To test this, we compared the current study to a study already completed using ATS ERS 2005 in a similar asthmatic population. When comparing within-visit variability of two FEV1 tests conducted 30 minutes apart, we noticed a reduction of the within-patient average variability from 83 milliliters or 4.9% with the old 2005 standards to 49 milliliters or 2.4% in the study using the new 2019 standards. Provisional data also shows similar magnitude of reduction when considering coefficient of variability of on-therapy treatment responses.